session is LaBella, and she's going to talk about population structure and migration access using 45 discordant gene regions in the deep sea limpet and Limpet.
I got it, yeah. Thanks. Thanks for coming. So this is a little bit different from some of the biogeographical talks you've heard before. So I'm going to focus a little bit on biogeography, a little bit on population structure, and sort of try to synthesize it. So what I'm studying is a deep sea limpet, Lepidodrillus affinis strolli, and these lepidodrilled limpets are found in high abundance on deep sea hydrothermal vents worldwide. And so hydrothermal vents, as you can see in artist rendering here, are typically thought of as volatile habitats. They're frequently created and destroyed by catastrophic tectonic events, and they're also patchily distributed along the active hydrothermal regions along the continental plates. And so this is what we typically think of as a metapopulation. We don't expect to see a lot of differentiation between somewhat nearby populations. And so we had a question, is this true? Do we see this widespread connectivity between vent sites? So we have two main sampling locations, and so we're in the Oceania Sea, right off Papua New Guinea. The first location is the Manus Basin, where we had 2 vent sites. The second location off of the Tonga Trench is the Lao Basin. And so they're separated by about 4,000 kilometers. And we wanted to know, are we seeing this widespread connectivity that we thought we might find in this region? And this is of particular interest because the vent sites in the Manus Basin are being developed for mining, and which may end up destroying these locations. And we wanted to know, is the connectivity high enough to repopulate these regions? So the next question was, what kind of genetic data did we want to collect? And what we ended up using is the sequencing of exon-primed intron-crossing PCR products, and this method was modeled off the work of Dr. David Weitzrath, who basically said, go for the most number of loci you can try and capture. And there were 3 main reasons that we went with this kind of technique. And the first is that we could target loci using a transcriptome, so we didn't need an assembled genome, which we didn't have for our species of limpet. And we did attempt to choose single copy genes a priori, and that will come in importance later. The second is that we can physically link these SNPs within the individual haplotypes. And this allows us to do sort of old school phylogenetic techniques like build gene trees, but also capture enough variation, hopefully, that we can start to do more widespread SNP analysis. So we wanted to kind of get the best of both worlds, sort of the old school and the new school analysis. So just a brief explanation, because this is a time-consuming portion of this, is that we assembled a transcriptome from a single individual. This transcriptome enriched in mRNA was then aligned to a sample genome. This is the Latia gigantea genome that was used here. You can actually use a Drosophila genome as well. It works fairly well. Where you see gaps in these mRNA sequences are putative introns. And then we can design primers to the exonic regions, which are more likely to be conserved and work across multiple individuals and sometimes even multiple species to amplify putative introns. They're then barcoded, and we used 454 sequencing for this data set, but the error rate was so high that we're probably not going to use that for future analyses using this technique. So the benefit to this that we really liked is that we were able to physically link our SNPs within an individual. So when you assemble the introns from a single individual, you sort of see these polymorphic sites. But how do you get 2 haplotypes for every individual? And the way we did this was we actually physically linked the SNPs using the read data. So all of the reads that didn't cover more than 1 SNP were eliminated, and you can actually physically build haplotypes for every individual from these shorter reads. So this works a little bit more like the typical Sanger sequencing, but we can do it with high throughput data and get 2 haplotypes per individual that are phased using physical linkage and not a statistical package. So what did we get when we did this analysis? We got 36 gene regions, and there were 45 loci within that, so sometimes we got an exon and an intron from each of these gene regions. And we had about 38 individuals per vent per basin. They ranged from lower to higher. And so what I was saying before is that we're able to do multiple types of analysis. So we can build phylogenetic trees using PALBSTAR. We can do a coalescent analysis using migrate-N, and we can also look at a clustering algorithm-like structure. So what did we see? The big story is we did see differentiation between the two basins, which was not what we initially expected. But even though we saw differentiation, we could detect low levels of migration between the two basins. And this is likely due to stepping stones, but we don't have data from the vents that exist along the axis where these two are connected. More interestingly, we saw different signals across all the different loci. So like you were saying, there's lots of different loci that you can look at, and they actually don't all say the same thing. But that's not necessarily a bad thing. What we think we're seeing is the effects of gene flow impacting different loci throughout the genome. So I'm going to show you a sample real quick of what we saw in these discordant loci. We have 3 examples. They're a thyrodoxin-like exon, an EIF2 gamma intron, and an EIF1 alpha intron. I'm going to show a phylogenetic tree, then some sample migrate end results, and then some sample structure results. And this is just the number of SNPs that was in each of these loci. So the first example we saw in this exon was a large clade exclusive to the Manus basin. And so what this suggests is that we're not seeing gene flow out of this clade from the Manus basin. So we might say, okay, it's predominantly allowed to Manus gene flow. But then when we look at the second gene that I have an example of, EIF2 gamma, we're seeing large predominant Lao clades, which suggests the opposite from what the first gene said. So then what happens when we look at the third gene? We see near reciprocal monophyly with a single individual at the bottom who has one haplotype that clusters within the Manus basin. So this suggests that while they're highly divergent, there is some Manus to Lao gene flow. So what do we take from this? This is just one type of analysis. We can do another type. So then we have different migrate end runs with different parameters, both maximum likelihood and Bayesian. And what you can see in the first instance is a predominance of Lao to Manus gene flow. The second instance, a predominance of Manus to Lao. And in the third, a lower level of overall gene flow. These are all scaled the same. And a slight peak in the Manus to Lao, which that single individual suggested. Okay, so we can do even more analysis with this type of data and do a traditional structure analysis. Structure works better if you actually combine all the loci, but this is just a simple clustering algorithm to see within each of these loci, are we detecting some structure? And in the first instance, you have this predominantly pure Lao group, and a little bit less so here in the Manus, and then here, of course, you have a very strong structure, except for this single individual here, which corresponds to that individual. So within each of these loci, what we're seeing is sort of a consistent story across all of the different analyses, but then across the board, how do we make sense of these discordant loci? Well, good thing for us, we didn't just do 3 genes, we did 45 loci. So what you can do, and this is just a sample, I didn't make all the figures look very nice for you, but we can start to divide this up into what it looks like across all these 45 genes. And so anywhere where you see red is a predominant Lao to Manus result, anywhere where you see blue, Manus to Lao, and anywhere where you see purple is a combination of both. And this is just 2 different types of structure analysis and 2 different types of migrate analysis. And what you can see is in the middle you see a lot of genes with either little signal or sort of mixed signal, but on the ends you see some loci that show strong directionality in either direction. So this is sort of the first pass where we looked at each of the loci independently. But we can also start to group them into groups of 15 or look at them all together to get a bigger picture of what's going on. And so when we combine the sets of 15, we lose a little bit of the signal because they are variable. They weren't divided by this group shows Lao to Manus, this group shows Manus to Lao. And so we do see in the first set a predominance of sort of Lao to Manus and Manus to Lao. We lose some of that structure, but it's still predominantly there. So what we can conclude is that we are seeing some low level of migration and gene flow between these 2 sites. What we're trying to assess more accurately is what the amount of that is and whether or not we would likely see recolonization of the Manus basin. But because there's likely stepping stones, more likely than not, we would see some recolonization of the Manus basin from either the Lao basin or some of the stepping stones in between. So in summary, what we see is that with 45 loci, we are capturing enough variation to use both the more traditional coalescent analyses and then also some of the larger clustering and techniques that come with, rad data or more widespread SNP data. And we are detecting this bidirectional migration in these differentiated populations. And this method, it is a little bit high labor. I'm in the process of repeating this for another species. It's a lot of PCR, but it is relatively low cost. You're going for high impact portions of the genome to try and maximize the variation, but minimize the total amount of data so that you have some concept of what you're actually looking at. And what we need to do in the future is we're trying to sort of look at, like I said, the magnitude more carefully, and the time scale. Is this ongoing, or is this something that is not as modern? And then we're also trying to repeat this and model these discordant loci. Can we see this in a model when we simulate the data? Are we seeing imprints of a low-level migration accumulate in different parts of the genome? So with that, I'd like to thank everyone who supported all of the data collection. Talk about expensive data collection. The DeepC is not cheap and easy to get to, so we do need to maximize our analysis. Thanks to Nautilus Minerals for support for the crews and all of those that helped produce all of this data and analyze it. And with that, I will take some questions before we go get coffee. Yeah.
Invested in next-generation sequencing stuff. Why are you opting for PCR rather than long-read sequencing such as Bio, for instance?
Yeah, so there's, the question was, why aren't we going for sort of the longer PacBio sequencing? And part of the reason is that we really don't have a very good reference genome for other species. So we got lucky with this region that we actually have a similar limpet genome, but I'm working on other species where we don't have anything anywhere closely related to it. So it's nice to be able to look across these different organisms. We are hopefully going to do actually PacBio sequencing on the PCR products, but like I said, it's sort of that